Hey folks, this course is divided into 4 quarters, and each quarter deals with considerably different content. So the first quarter deals with chemistry and biochemistry. The second quarter deals with the cell and its parts and what the parts do. The third quarter deals with metabolism and in particular catabolism, and the fourth quarter deals with cell division and genetics and what we could collectively call molecular biology. Each of those topics, cell division, genetics, and molecular biology, actually relate to one another. So it is in fact a fourth unit of content that we will now begin. We will start with cell division. Your textbook actually splits cell division into 2 parts, chapter 12 and 13, which mitosis and meiosis. So this is the first lecture on cell division, and this is the first part of that lecture. Let's proceed. We'll begin with this question: where do cells come from? Well, if you said other cells, then that would be a good answer. In our contemporary world, now, cells will only come from other cells. Okay, this question was not intended to, like we did at the beginning of the course, what is the origin of cells? Did they begin on Earth? Did they come from space? And that sort of thing. I mean, that is another question one could ask. Which does not have an easy answer, but this actually has a very easy answer. In our contemporary context, cells only come from other cells, and the process that produces them is cell division. We can think of the life of a cell as being formed by cell division. Then that cell grows and divides itself, right? So that cycle, if you will, beginning with the birth of the cell from cell division, growth, and cell division, that would be, we could say, the life of a cell. Do all cells divide? I'm asking. You have to offer an answer. So actually, no is the correct answer. If you are aware, or if you know what neurons are nerve cells, these cells don't divide in our adult bodies. They once divided, and that's what led to our having so many neurons. We, have billions of neurons, but they no longer divide, right? So we have to qualify, or at least recognize that all cells do not divide. Some do, but not all. And however, all cells were produced by cell division. Since we're talking about cell division, why don't we answer this question? Why do cells divide? There's actually multiple answers, as you can see. One reason is for growth. In a multicellular organism, an individual grows by cell division. At least that's a big part of the growth of the organism. Most multicellular organisms also develop. They're not simply a mass of cells. They have different cell types, different tissues, different organs, and so growth and development both involve cell division. Occasionally, a tissue or an organ will become damaged. At that point, cell division could very well serve to repair that damage. Good as new. There are other tissues in our body that we will constantly regenerate, constantly renew, because it's just a good idea, right? Certain tissues deal with a day-to-day abuse, kind of a constant abuse based on their function, right? The skin is a good example. It's exposed to the elements, it's exposed to temperature, it's exposed to radiation from the sun. A lot of insults happen to our skin, and so it's just a good idea to constantly refresh and renew the skin, and so we do. And that's done by cell division. Reproduction inevitably involves cell division. And I don't care if you're a single-celled life or if you're a multi-celled life, you reproduce by cell division, or it's part of your cell division, or part of your reproduction, excuse me. So here's 3 reasons why cells divide. Yeah, and let's just say if you are a single-celled organism, this is the only reason you divide, right? When you divide, you reproduce. So point 1 and 2 don't apply. All right, so with that, let's commence our discussion of cell division. And we will now proceed to focus on chromosomes more than we have at any other point in the course. I mean, we've done a cursory sort of nod or mention at best. Now we will really begin to scrutinize the chromosomes and must in order to understand cell division. So chromosomes and cell division, let's begin. We have a lot to talk about in this chapter and 2 chapters actually. We'll talk about cell division in bacteria, then go on to talk about cell division in eukaryotes. A unique aspect of cell division is the cell cycle. Cell division is actually part of the cell cycle. And this wording, the cell cycle, is unique to eukaryotes. And that's why I put it in blue. Like all terminology in the course, the blue indicates it's a term and it's reserved for euks. We'll then go on to talk about forms of the genetic material. As I said, we're going to talk about chromosomes more, but what is chromosome exactly? What does that mean? What is chromatin, if you're familiar with that term? We mentioned it earlier on. I mean, how does DNA relate to, either one of those terms? I mean, what are genes for that matter? Something else we've talked a little bit about, but we need to begin to talk more about now. Let's get into it. We'll then go on to talk about mitosis. That concludes chapter 12, and then go from there to chapter 13, where we'll talk about meiosis. Let's begin with bacteria, which are the main type of prokaryotes. Most prokaryotes are bacteria, and how cell division operates there. All bacteria are single-celled, by the way, single-celled life. Most have a single chromosome, just one. And the shape is what? Circular. It's a circle. It's a circular DNA molecule with no ends. And what we might say, covalently closed circle. Okay, to tie it back to chemical interactions we're aware of. Circular DNA. And that DNA is copied in the process of DNA replication. Again, a terminology. That's the name of the process that copies DNA. Cells must copy their DNA before they divide into two cells. If they don't, one of the two daughter cells will not have the DNA, and the life of that cell is at an end. Cells depend on DNA. As you know, it's the code. It's the code that makes the proteins of the cell. Indispensable. So there's actually a order of operations here, isn't there? DNA replication, cell division. DNA replication, cell division. Those two processes always go hand in hand, DNA replication and then cell division. That's a topic that's taken up in chapter 16, and we'll be there before you know it. In bacteria, cells divide in a process by which they lengthen, and then they split in two. That process is called binary fission. Again, the cell lengthens and it splits in two. Let's be clear, that is not mitosis. That is not meiosis. It's binary fission. It frankly is a simpler, mechanically simpler process. And one way to think about it is, as long as bacteria has adequate nutrients, they continue to divide and divide and divide and make more bacteria. Right? Maybe a little simplistic, but I think there's a lot of truth in that statement. So this is an electron micrograph showing binary fission. You can actually see the cell has lengthened. The rod-shaped bacterium, it has lengthened, and then it is split in two there in down the middle. As usual, the micrographs are accompanied by diagrams, which kind of help you to see what you're supposed to see, get the important idea. And so again, this is a rod-shaped bacterium, and here is its chromosome. So recall, the chromosome is a circular molecule, right? If you were to start at one point on this circle and then just follow it all the way around, right? This may not be so easy. You might need to actually enlarge this figure. I probably do. All right. The point is that you should get back to the same point where you started, okay? Because that is a circle. Now the cell is getting longer, right? Ooh, also the DNA has been replicated. All right, so what did we say? DNA replication and then cell division, and then cell division, and then cell division. And notice when the cell divides, it divides in such a way that each daughter cell gets a DNA, which again is the information to make proteins, the information to run the cell. Bacteria. Eukaryotic cell division is more complicated. Why is that the case? Well, frankly, there's more than one answer. Here's a diagram of a eukaryotic cell we looked at earlier, right? So one of the things you should recognize immediately is that eukaryotes have they're larger cells, all right? But I wasn't going to make a point about that, but they contain internal compartments. Bacteria contain internal compartments, and that means the cell has to deal with that, has to contend with that when the cell divides. The other thing is the DNA. There's usually multiple chromosomes, right? You can't necessarily see that here, but in fact, there's usually multiple chromosomes. So let's address both these points. The membrane-bound organelles. There are numerous membrane-bound organelles in a eukaryotic cell. So how does the cell deal with those when it divides? First, recall that mitochondria and chloroplast are semi-autonomous, right? Mitochondria and chloroplast will divide on their own in a fission type division, right? So that takes care of them. If these organelles divide, then we'll have more of them, and then when the cell divides, then both daughter cells will get, ample mitochondria and chloroplast, so they retain those organelles. The other organelles, ER, Golgi, NE means nuclear envelope, not semi-autonomous. Right? These are not like mitochondria and chloroplast, but do recall, they're all part of the endomembrane system. Oh yes, ER and Golgi and lysosomes and even the nuclear envelope, all part of the endomembrane system. So they share that in common. Remember that ability to pass membrane around between them, send vesicles between them. So what fate do they face? They will disintegrate. All of these structures disintegrate. They come apart. The cell divides, and each daughter gets some parts. Those structures then proceed to reassemble in the daughter cells. A new ER appears in both daughter cells. A new Golgi appears in both daughter cells. New nuclear envelope appears in both daughter cells. So it's a process of disintegration and then re-entry in the daughter cells. All right? That takes care of the organelles, then chromosomes. Again, eukaryotes have multiple chromosomes, and I mean, therein lies the challenge. Right? So you have multiple chromosomes, not just the one. How do you make sure that all of those chromosomes not only get duplicated, but then get segregated to the daughter cell so that each daughter gets a one of the chromosomes? Right? Well, to be continued. That's a topic we're going to continue to talk about. Recall that there are two types of cell division in eukaryotes. You know what these are, mitosis and meiosis. Mitosis produces cells identical to the parent cell. Two cells identical to the parent. What about meiosis? Well, recall, you get cells with half as many chromosomes. Number of chromosomes becomes half. That by tells you something immediately about the number of chromosomes in cells that do meiosis. What does it tell you? Okay, let me say it again. If meiosis halves the number of chromosomes, what does it tell you about the number of chromosomes in cells that do meiosis? It must be an even number. You can only half an even number. I'm just saying. We said growth and repair, regeneration were all functions of cell division. In fact, they are accomplished by mitosis. Mitosis is what produces more cells for these purposes. Then what the heck does meiosis do? It's exclusively for reproduction. It's a special cell division for the purpose of reproduction and no other purpose, actually. Again, we'll start with mitosis, and so we'll focus on that first. Mitosis happens in all of the cells of your body. Now, let me qualify that. All of the cells of your body. All of the cells of your body are produced by mitosis. In your adult body, not all cells are doing mitosis. We said the neurons. Remember the neurons not doing mitosis, but they did do mitosis. That's why you have billions of neurons, because they did do mitosis. All right, so mitosis, we can say, happens in body cells. cells of the body. Cells of the body are called somatic cells. So the cells in your hand are all somatic cells. The cells in your nose, all somatic cells. The cells in your brain, all somatic cells. What is not a somatic cell? All right, the sex cells. The sex cells, the egg and sperm, and actually the cells that become egg and sperm are also not sex. I'm sorry, they're not somatic cells, right? So it's everything but the sex cells, the egg and the sperm and the cells that become them. All right, the next topic is the cell cycle. Once again, this is a process unique to whom? Eukaryotes. You got it. The cell cycle. Never, ever, ever hear a microbiologist say that, because that doesn't apply to bacteria. What the heck is the cell cycle? It's a tightly regulated series of events that culminate in cell division. Tightly regulated series of events culminating in cell division. And it is unique to eukaryotes. Now, knowing that definition, we may conclude that this process is controlled by many what? I'm just going for enzymes. You realize, I think, at this point that the cell, the chemistry of the cell is run by enzymes. The chemistry is accomplished by enzymes. Therefore, enzymes must be involved, right? I mean, how else would you have a tightly regulated series of events? I mean, it's got to involve enzymes. Well, good answer. If you said that, and if you didn't, just be aware that that's what's going to happen. The text gets more into the enzymes. In fact, figure 17 in this chapter talks about it, but you are not responsible for that. You will pick that topic up in cell biology when you take that down the road. I would be remiss if I didn't say that the cell cycle is, in fact, the most intensely studied process of biology, or among the most intensely studied processes. Cell biology. Why is that? We just love cell division. Right? We're so interested. I mean, well, it is an interesting process. I'm joking. It's not just because we're interested in cell division. No, there's a disease, right, that actually results from too much cell division. Right? Of course, talking about cancer. Cancer and heart disease are the number 1 and 2. They're really close. They're always kind of neck and neck. Causes of death in the U.S., to be sure. It's a huge reason that people die is oftentimes because of cancer. So we're interested in knowing about cancer and knowing how to treat cancer, and how to we'd like to beat cancer, right? Is sometimes what you hear, but it's a real. I mean, this is not a battle. This is a war. I mean, this is really cell biology and cancer go hand in hand, like I don't know what does. Where you have cell division, you have the propensity for cancer, right? They're just two peas in a pod. They go along together, and this is part of the challenge, I think, in treating this disease. That's why people study cell cycle. Cell division, as I said earlier, is part of the cycle. It's part of it. And it happens in what we would call the M phase of the cycle. Obviously, M is for mitosis. Right? So what is the rest of the cycle apart from M phase? Well, the rest of the cycle is the phase or phases between cell divisions, between divisions. So we call that interphase, the portion between cell divisions. And frankly, that's most of the cell's life. 90% of the cell's life is interphase, only 10% then the M phase. But that said, interphase has 3 parts or subphases. And here they are. S phase is the phase when the chromosomes are duplicated. Excuse me. Chromosome duplication happens in that phase. So we have a name for it when that happens, as we said, before cell division. Why is it S? Well, it's for synthesis, DNA synthesis of the DNA. There are 2 phases separating M phase from S phase. All right, so it goes M phase, a gap, and then S phase and another gap. You see what I mean? M gap, S gap, M gap, S gap, and so on. So we call those gap phases. Why not? The phase between going from M to S, that first gap is called G1. Then going from S M, that second gap is called G2. So we have G1 and G2. So they separate M from S. Growth of the cell will occur during the gap phases. Another thing that happens is the cell will prepare for the upcoming phase. So in gap 1, the cell gets ready for DNA replication. It's ready to do that. In gap 2, the cell gets ready for cell division. Gets all its ducks in a row, so to speak. All right, so we're always preparing for the next phase in the previous one. Part of that preparation in G1, in G2, is to carefully inspect the nuclear DNA. Both gap phases will do this. Why do they do that? The reason is that if there are problems with the DNA, it's like some kind of damage, that happens, DNA damage, and you go on to do the next phase. Let's say you go from G1 to S. You do replication. Then the damage can become a permanent change. A permanent change in the DNA sequence. There's a name for permanent change, permanent in DNA, and that's called mutation. Oops. But the answer was going to reveal, but I guess I'm just telling you the answer verbally. Right, so the reason why this is done is really to prevent mutation. To prevent mutation, to prevent a permanent change in the cells. So again, mitosis happens at the end of the cell cycle, right? So we have G1, S, G2, and then M. M phase itself has 2 parts. We have the division of the nucleus or the contents of the nucleus. That's called mitosis. And then we have division of the cytoplasm. That's called cytokinesis. So mitosis and cytokinesis are actually two separate things that go on in the M phase. The whole cycle is shown here. Let's start with cell division. We said that the life of a cell kind of begins with cell division. So here it is. The cell is born, right? This is its sibling here. We call them daughter cells. All right. And we hit G1. G1. G1 means growth. As the figure indicates, this is actually the primary growth phase. Most growth happens here. And the cell basically gets to a normal and functional size. A normal functional size. This is how long it takes to replicate DNA in S phase. Yeah, it is substantial. Hours, right? If this was a human cell, this would be hours of time here. G2 is additional growth. G2 is additional growth for the purpose of cell division. Right, when a cell divides, becomes 2, then immediately it becomes half as large. So we got to have some additional growth so we're not too small when the cell divides. In adult organisms, only some cells divide. Yeah, we said this earlier. Which implies that a cell can stop dividing. It can leave the cycle and stop dividing. And at this point, it's in what we might call a non-dividing state. We need a better name than that. Let's call it G0. G0 phase. A non-dividing state. So here's a question. Knowing what you know about the cell cycle, where would you want to exit from? Somewhere here, G1, from S phase, or from G2? Well, I hope you said G1 because that is the right answer. What did we say? The cell grows to a normal size, a functional size. And that's what we need if the cell is to perform a job in the body. The only reason to go on to S phase is if you're going to do what? Go on to M. The only reason you replicate your DNA is if you need copies, because you're going to divide the cell. Don't do S phase unless you're going to do M phase. And again, G2 is extra growth, we said. Extra growth for cell division. So if you're going to just be a cell, not make more cells, leave from S phase, leave from G1, excuse me. Leave from G1. Leave from the end of G1, I would say. Near the end of G1 where you've grown to a good size, and then leave. Okay? Again, some cells divide, but some cells don't. And how quickly depends on the cell type. We can categorize cells into 3 categories of frequency. Those cells that divide continuously, those that divide based on need, and those that rarely or never divide. Again, skin cells are a good example of number 1. The cells of your GI tract, another example of number 1. and frankly, the cells of the blood. Your blood is produced by bone marrow, and those cells constantly dividing. You're constantly making new blood cells because they're so important for your body. You always want fresh supply. All right, so what do we say? Skin cells, gut cells, lining of the gut, and blood cells, always dividing. Some cells will only divide when you need more of them. And textbook example is liver, right? So if you consume alcohol daily, right? Consume. I mean, really what I'm getting at is if you're an alcoholic, probably don't consume alcohol daily. Let's say that. Okay, that's too much. I think in many cases. If you are an alcoholic, what's your liver like? Is it smaller or is it larger or is it the same size as a regular person? Larger. Yeah, it's a larger. Alcoholics have a larger liver. And the reason for that is, recall, liver is involved in detoxification. If you're consuming a toxic substance, ethanol is toxic to the body, and if you're consuming that, then you do need to more liver to detoxify that ethanol. All right, so why do you have more liver? Because of cell division. You had a need for liver, and cell division made it for you. All right. I shouldn't call non-alcoholics regular people. That's not nice. But it is, alcoholism is an unfortunate disease and a serious one, and so I think abstain or moderation would be advised. All right, of the day. Some cells divide rarely or never. Neurons. Neurons were an example. Muscle cells are another example. Your muscle cells do not divide in your adult body. And sometimes students will ask me, well, how come when you work out, your muscles grow? That's not cell division. No, you're not growing more muscle cells because you're doing bench press. You put more protein into your muscle cells. Right? You gotta, by the way, if you ever try to build muscle mass, you better have a protein-rich diet. Those two go hand in hand. Protein consumption, weight training. Protein consumption, weight training. Protein consumption, weight training. Or you'll never ever, you might tone up a bit, but you won't really see a gain of mass. Category 2 and category 3 both involve cells being in G0. In category 2, the cells are in G0, but they can come out. They can come out of G0 and resume the cell cycle. Resume the cycle. Make more cells. Resume the cycle. Category 3, permanent G0. Neurons, muscles, permanently in G0. They cannot divide again. They're what we might call post-mitotic cells. And this is why, say, CNS injury, spinal cord injury, is so such a traumatic type of damage to the body, because you cannot regrow cells of the central nervous system. Okay? You can't repair or regrow those cells, and so paralysis can be the unfortunate result. G0. Most human cells are in G0, by the way. So at any given point in time in an adult individual, most cells are in G0. In fact, there's only a few we said are constantly dividing. All right, we'll pick it up there with the forms of the genetic material. Okay, this is the second part of today's lecture. Let's go ahead and continue and talk about the various forms of the genetic material. So this is a fluorescent micrograph or series of micrographs, basically photographs taken with a microscope, in which something has been stained in the cell. What has been stained? So you're just looking at the DNA. You can see the shape of the nucleus here and here, although that lovely round shape kind of goes away for some reason. And here you can actually see, well, you can see chromosomes. Some reason you couldn't really see that early on here. Maybe something here, but here. Well, we can see something here. You can see mitosis going on here, right? And now I don't know what that is, just some fuzz. And yeah, so we end up with a nucleus again, right? Although it's a kind of a smaller one, maybe about half as large as it was to begin with. All right, so this is actually M phase. We're looking at the events of M phase of the cell cycle, but specifically what happens to the DNA. And one thing we can conclude is that the eukaryotic chromosomes can become more or less compact. That is why we do not always see them. If we see them, they're compact. If we don't, they're less compact. Does that make sense? So how can we change how compact they are? Well, recall, which we said way back at the beginning of the course, that DNA is wrapped around protein. The DNA in the nucleus is wrapped around protein. I want to say chapter 2. We were talking about charge at the time. This complex means different molecules held together by non-covalent bonds, held together by weak interactions. Complex. This complex of DNA and protein is known as chromatin. And why? Why must it be with protein? Well, please remember, I'm sure you've heard this before, that one human cell contains 6 feet of DNA. That's one cell. And in fact, it's one nucleus. One nucleus of one cell has 6 feet of DNA. That's just me. I'm about 6 feet tall. And so if you could stretch a thread from the top of my head to the bottom, I mean, that's one cell's DNA. So that DNA must be compacted to fit into one cell, into one nucleus. And remember, this is accomplished, at least in part, by physically wrapping the DNA around protein. Okay, this is not a figurative description. This is not some kind of abstraction. This is a solid, actual thing. The DNA is wrapped almost twice around protein. It's every 200 base pairs. Every 200 base pairs, you have one of these wrappings, right? With these little beads with the DNA wrapped around. Right. So what we're looking at here in that first cell, in the nucleus, is chromatin. It's a complex of DNA and protein. That's all we can really see, right? This is the DNA and the protein, and we call that chromatin. This is chromatin. This is chromatin here as well. So what is happening again? Is the DNA becoming less compact or more? More. More is the answer. It's becoming more compacted, as we proceed, which is why here you can see individual DNAs. Right? So they were there to begin with, but they're compacting so you can see them. Let's make that a key concept. During M phase, chromatin packs so tightly that the individual chromosomes become visible. They're there to begin with. They become visible in M phase. So this is chromatin. This is what you have in interphase. But this becomes compacted here and looped and coiled into, there it is, into this structure here and here. This is a chromosome. And notice that is a chromosome. What is it? It's DNA wrapped around proteins and compacted to a great degree. Yeah, so here's an actual photograph of a micrograph of chromosomes, as you've seen before. This figure actually comes from another Bio 1 textbook, but your figure or your text has such a figure in number 21. Oh, I'm sorry, chapter 16, figure 21, 1621. That's where I think you should see a comparable figure, if you like. So here, right here, we're seeing that compacted DNA. The DNA has been compacted. Why? So that we can separate, so that the cell can separate the DNA into two resulting cells, daughter cells. So what is a chromosome anyway? A chromosome in eukaryotes is one DNA molecule. One DNA molecule plus the associated proteins. Again, it's wrapped around. All of that is a chromosome. In eukaryotes, the DNA is linear, not circular. It's linear. So you got two ends. Ends are called what? Telomeres. Telomeres. The ends of a chromosome are telomeres. DNA has two telomeres. And a chromosome in eukaryotes contains hundreds to thousands of genes. Hundreds to thousands. Not millions, not 10, hundreds to thousands. It depends how long the chromosome is. A small chromosome, maybe a few hundred, a long chromosome, a few thousand. Another key concept, each eukaryotic species has a specific number of chromosomes. For example, human cells have how many chromosomes? No, this is one of those answers you want to know, the numbers you want to have memorized. 46. Human cells have 46 chromosomes. That's what we mean when we say one cell has 6 feet of DNA. That's the 46. If you could stretch the 46 chromosomes end to end to end to end, you would have 6 feet of DNA. Same thing. So the 6 feet is in 46 pieces, isn't it? So what is this structure? As we said, that's a chromosome. This is now a very high magnification. I think this is a scanning electron micrograph, SEM. But recognize that this is more accurately referred to as a replicated chromosome. It's a chromosome, but it's a replicated chromosome, meaning it's the product made for the purpose of cell division. What did we say? Replication, cell division. Replication, cell division. Every cell division is preceded by replication. Replication copies DNA. This is the result. There's one DNA here. There's one DNA here. That is one DNA on the left. Highly coiled, highly compacted. And another DNA on the right, again, coiled and compacted. We need a name for just one of those, I think. And that we will call a chromatid. So a replicated chromosome consists of two chromatids, also known as sister chromatids. Make that a key concept. A replicated chromosome has two sister chromatids. Each one is one DNA, or contains one DNA, again, wrapped around protein. The DNAs are the result of replication, the copying of DNA. They're therefore identical. The DNAs are identical. And each chromosome has a centromere. Centromere. What does centromere mean? Well, it's this region here. The centromere is where the replicated chromosome, the sister chromatids, are going to be pulled apart from. Yeah, so what's going to happen in cell division, please remember, a structure called a spindle forms, and the spindle grabs hold of the replicated chromosome at this location. It grabs hold from both sides, where I'm showing my pointer here, grabs hold, and pulls and separates the sister chromatids. Once they're pulled apart, the sisters disappear. Well, they seem to disappear, but actually what they do is they uncompact. They become less compact. Another way to say that is when they appear, they condense, and when they disappear, they decondense. Condense, decondense. So your Campbell, always with the strong figures, puts it all, or summarizes it all into one figure here. So here we have a cell prior to cell division, and I guess this is the simplest possible cell. It has just one chromosome. Well, one chromosome, remember, each chromosome is a DNA molecule, and so that's going to be DNA, right? This must be this, right? This is a little bit of artistic license here, because if this is in fact interphase, which I know it to be, the chromosome would not be condensed. I mean, this is a condensed structure. It would be decondensed. It would be spread out in the nucleus. All right, so this is an artistic license here, but I think it helps to make a point. All right, so before cell division, there's one DNA. One chromosome means one DNA. In preparation for cell division, the DNA is copied. The DNA is copied. It's called DNA replication, and it produces two identical DNAs. That happens in S phase, and that's the result. Right? Each chromatid is a DNA. Chromatid on the right, DNA on the right. chromatid on the left, DNA on the left. Okay? The process of cell division then separates the sister chromatids. In other words, the DNAs. Oh, again, that structure we saw on the slide, previous slide, is that, right? Make no mistake. Ah, there we go. In M phase, the cell divides, and again, mitosis and cytokinesis. Mitosis will separate the sister chromatids. Cytokinesis will separate the cytoplasm or split the cytoplasm. And we get two cells. We get daughter cells, which are identical to the parental cell. Since the parental cell had one DNA, the daughter cells likewise have one DNA each. Okay? I asked you for a question. I didn't hear anything, so I have a question. If 90% of the cell cycle is interphase, what does a nucleus usually contain? Visible chromosomes or chromatin? Chromatin, good. Usually it contains chromatin, 90% of the time, chromatin, which are the chromosomes in a diffuse state, a decondensed state. They're in a chromatin state, if you will. One more. Genome. Genome. Oops, what did I do? A genome is the total genetic information possessed by an organism. All the genetic information. But make no mistake, you don't have to go to every cell to find, combine all the cells. That's not what it means. All right? Where do you find the genome? In each cell. Each cell. Each cell has the total genetic information. Each cell contains the genome. Think about that. The human genome has a little bit above 20,000 genes. Right? That means every cell has 20,000 genes in it. The total genetic information for humans, to make a human, to run a human, 20,000 genes. So these terms, they all are related, but they have different meanings. So please, know how they relate, but know how they are different. Genes, I think we didn't dedicate section 2, but we've talked about them before. We'll talk about them more. A gene is a region of DNA. Remember, a gene is a region of DNA that is used to make a protein. And as we said, a chromosome will have hundreds to thousands of genes depending on the size of the chromosome. All right, let's pick it up there in the next segment.